Welcome back, everyone. This is Doug McConnell speaking. I am the co-founder of a nonprofit called A Long Swim that is dedicated to using open water and marathon distance swimming to raise money for collaborative ALS research. We're delighted this evening to really show off some of that collaboration and so on. Of course, the star of the show is Hande Ozdinler, my friend and really inspiration for so much of the swimming that we do. Dr. Ozdinler, of course, is an associate professor, the Department of Neurology at Northwestern University's Feinberg School of Medicine right here in Chicago. And is also on the faculty, among other things, of the Chemistry of Life Processes Institute, that is a collaboration within the four walls of Northwestern itself. Dr. Ozdinler's research really has focused and pioneered, I think, the work in upper motor neurons as it relates to ALS and motor neuron disease. We were delighted when she agreed to be the scientific advisor for A Long Swim. We're having this webinar at 6:30 on a Friday evening in Chicago, which is terribly awkward for most. But one of the reasons that we did that was because our visitor, Dr. Steve Vucic, he also responds, we've learned, to other pronunciations of his name, is joining us from Sydney, Australia. Dr. Vucic is the professor of neurology. I'm reading this to make sure I get it right, of Western Clinical School as part of the University of Sydney, which also includes Westmead Hospital and Westmead Institute for Medical Research. Interestingly enough, Dr. Vucic trained for part of his career at Mass General in Boston, part of Harvard Medical School, of course, which also has Hande as one of its graduates as well. Dr. Vucic's research is very interesting, particularly as it relates to ALS, also known as motor neuron disease, and how the early sort of detection and intervention can be done, specifically as it relates to tonight's kind of naughty title of cortical hyperexcitation. I know that Dr. Vucic and Hande will both have very interesting commentary about this. Anyway, before I get way out of my element, Hande, please take it away.
Thank you so much, Zach, for the introduction. And I think Dr. Vucic is one of the pioneers, and his group is also one of the pioneers for the cortical component of ALS and how cortical hyperexcitation occurs very early in ALS. And I would like to welcome him to our program, to our webinar, and joining us from all over around the world, from Sydney. And happy Saturday morning to you. Thank you so much for coming. And we are delighted that you are here.
Thank you very much for inviting me. I'm absolutely honored. Good morning or good evening, Doug, Hande,
good morning. That's fine.
All your audience, and thank you for accommodating me today.
Of course. So, would you like to tell us a little bit about your research and also the accomplishments of the Sydney group? Because I know you play a very important role for the cortical component of ALS, and I would like to give the platform to you. Why don't you tell us more about your research, your team's research, and how important that is within the context of ALS pathology?
Steve, before you start, excuse me, I'm sorry to interrupt. I forgot one of the most important things, and that was to instruct our listeners and viewers about how to ask questions. Forgive me, but I know that they'll have some good ones. In the bottom of your screen, there is a Q&A box on the Zoom screen, and we're going to try to gather questions right in that Q&A box. So as questions come up, don't feel like you have to wait till the end of Dr. Vucic's remarks or Hande's remarks. Just load them up and take it from there. Anyway, Steve, forgive me.
Thank you very much, Doug. Absolutely. What I might do is I might start sharing my slides, if that's okay. I'll share my screen. And yes, this should be an interactive webinar. I'm very open to questions. Can everybody see my screen?
Yes.
No, you're good, Steve.
So thank you very much. Well, look, as you all know, motor neuron disease, ALS or Lou Gehrig's disease, is an extremely complex and terrible disorder. But one of the things that characterizes it is involvement of both upper and lower motor neurons simultaneously to variable degrees. In its typical or classical form, the ALS, which Charcot recognized, there's a combination of upper and lower motor neuron signs producing specific clinical features like brisk reflexes, spasticity, and progressive muscle wasting and twitching, cramping, and so on. In most patients, the disease starts in a limb, like an arm or a leg. In a smaller proportion, it starts in their throat with difficulty in speech and swallow. And really, one of the great thinkers of our times, I think, has to be Andrew Eisen, who's given a lot of thought to what is ALS, where does it begin, and how it evolves. And this is really a transformative review, and one that I've read many times, written by my colleague, co-written with my colleague, Professor Keenan, and obviously Professor Mitsumoto and Professor Swash, where it was really suggested that ALS is a disease that perhaps begins even at conception, and that factors during the growth of the fetus, the maternal environment, start to shape ALS. And then as one grows, there are a whole lot of what we call epigenetic factors, things that you meet in the environment as well, that kind of predispose you or push you towards ALS. And by the time we see ALS in sort of what I would call the prime of adulthood, mean age of 60, because I'm approaching that, one gets the expression of the disease with loss or degeneration of motor neurons. And for a long time, motor neuron disease was really classified as a neuromuscular disorder. And I will get onto the role of hyperexcitability and its role as a brain in one moment, but I'd like to just digress a little bit to just discuss a couple of papers on its pathogenesis. And supporting Andrew Eisen's and Matthew's review is this initial paper published by Amar Al-Chalabi's group, where they propose that ALS is a multi-step disease. So there's not one cause of motor neuron disease, but rather there are 6 steps. And I'm talking about the sporadic motor neuron disease. What these factors are, we can hypothesize, and there's various discussions on that, but there are 6 sequential steps that are required. And we've reproduced this in the Australian Caucasian cohort recently, and even more recently, a paper that's actually now been accepted in neurology in both the Japanese and the South Korean cohort. So irrespective of where you are in the world, whether you're of Asian background or Caucasian, you need about 5 to 6 steps, 5 to 6 factors to push you to develop motor neuron disease as we understand it. That is, to get muscle wasting and weakness and the rapidly progressive disease. And this is very important. And if you look at the genetic forms of motor neuron disease, various genes represent 1, 2, or 3 steps. For example, if you're born with a C9orf gene, you might need 3 steps. So genes don't cause motor neuron disease. They are very strong predictors of motor neuron disease. And this is where cortical hyperexcitability comes in. My strong feeling is that this is one of the steps, perhaps one of the final steps in a disease progression or disease evolution. And really, again, although Charcot observed the notion that both upper and lower motor neurons were involved in motor neuron disease, and he had some theories and perhaps hypothesized that it may begin there, although that's not definite. It really was Andrew Eisen, who I think was in a flight somewhere, that he came up with this theory called the dying forward hypothesis, where he suggested that and proposed that motor neuron disease is really a brain disorder. It actually starts in the brain, and that the corticomotor neurons, the cells that connect your brain to the motor neurons in your spinal cord, the nerves that control your arms and legs and swallow, etc., are the ones, these corticomotor neurons are hyperexcitable. And they are so hyperexcitable that they actually destroy the cell that they're meant to be stimulating, the anterior horn cells, through a glutamate-mediated excitotoxic process. Well, this was proposed in the early 90s, I think 92 or 93, and it really was revolutionary, garnering some really strong debate. But I think Andrew Eisen was correct. So, one of the ways we measure this is through transcranial magnetic stimulation. So we put a magnet on the top of the head and stimulate the brain by inducing an electric current. And we measure the size of the response that this produces. And this gives us an idea of how strong this corticomotor neuronal projection to a specific muscle is. And in the technique that we developed here in Sydney, rather than measuring the size of the response, what we measure is the current or the stimulus required to produce a specific response. So everybody has a certain level of excitation, amount of current that you need to put into the brain to get a response. In patients with ALS, you need less current to produce the same response, and that's called hyperexcitability. And the specific technique that we developed was a threshold tracking technique. This was adapted from Hugh Bostock's axonal excitability techniques and a previous attempt that they made. And so what we did, we tracked a specific target or specific size of the motor evoked potential called the tracking target. And so when the response was, motor response is higher, we reduce the current, and when it's smaller, we actually increase the current. And so we're measuring the changes in the current. And this is what a normal person sort of looks like. You get a degree of inhibition. This is a recording from a group of controls. So if we study myself, Hande, Doug, we all have a degree of what we call inhibition. And that term is called short interval intracortical inhibition. And we know that this is mediated by cells in your brain, in your motor cortex. They act via a chemical called GABA, which I know Hande studied quite well. And so we can actually measure that in vivo and in live human beings, not in animals, not in rats, but in patients, very quickly over a period of 8 to 10 minutes and relatively painlessly. So I might pause there.
If I can ask you a question. So many patients ask, so this is very powerful technique, but let's say you don't have any symptoms, like then there would be, it would be very hard, let me say, for patients to go to clinic and say, oh, can I please check for my cortical hyperexcitability, right? So do you actually perform these studies on familial ALS cases, let's say, who has the mutation but hasn't shown the phenotype yet, or the genetic disposition, or it is within the family, and who gets to be tested for this cortical hyperexcitability? Because if you don't have any symptoms, you wouldn't go to a doctor or in a medical facility and say, oh, can I please check my cortical hyperexcitability, right?
Look, absolutely. I mean, this is a diagnostic aid like an MRI or like a blood test or like a nerve study. So if you're healthy and normal, you wouldn't go and have this test done. However, if you present with muscle weakness and wasting and twitching and cramping, and the possibility of motor neuron disease or Lou Gehrig's disease is raised, then this could be a very good diagnostic surrogate to your clinical examination, to your other tests like nerve conduction study and EMG, like your blood test, like your MRI, to help your neurologist with the diagnosis of ALS. Now, what we found consistently studying, and we've studied close to 500-600 ALS patients over the last 10 years here, 10 to 12 years here in Sydney, is that cortical hyperexcitability, as indicated by, in one way, a significant reduction in this level of inhibition, is a consistent and early finding in ALS. It occurs at a very early disease stage in patients with sporadic ALS, and also may precede the loss of lower motor nerves, which I'll show you in the next slide, in sporadic ALS, and also may precede the onset of familial ALS by about 3 to 4 months. But what is important is that this hyperexcitability is not seen in what we call ALS mimicking disorders, patients that have, say, muscle wasting and weakness that are due to other causes. So we can distinguish cortical hyperexcitability can help distinguish ALS from non-ALS with a relatively good sensitivity of over 70% in a highly selected cohort, and a very good specificity. So, we're fairly certain if you're hyperexcitable, if your inhibition is less than that number of 5.5%, or if it's absent, you've got a very high chance, a high likelihood of having motor neuron disease. So you really only need to study about 2 patients in this highly selected cohort, and it can enhance your diagnosis. Again, I'd like to reiterate, this is it should not be used as a standalone test like anything else, like MRIs, like blood tests, but rather should be used in the appropriate clinical context. And just to show this, we studied a group of sporadic patients who had absolutely no disease in their arm, and we looked at their arm in a number of different ways with various nerve studies and so on, and needle EMG, and we found that the development of hyperexcitability may occur for a number of months before the onset of lower motor neuron dysfunction.
This is very exciting, because can you imagine the cortical phenomena or pathology that you observe is much earlier than the symptom onset, and you see this in sporadic ALS patients. But my question to you is, yes, you use this technique or approach in Australia, but do you know if this is widely used or applied in other clinical settings in Europe, in the United States, in Japan, in China, in other places around the world, or this is very selective or mostly applied at your center in Australia?
Look, that's a very good question. At this stage, this is still a relatively boutique technique in the sense that it's mostly used in Australia. It's used in a center in the UK, and also in a couple of centers in Japan, mostly people that we have trained up. However, we are commercializing this, and we are in the process, hopefully by the middle to late this year, to roll out a device that will be an off-the-shelf device where patients can, doctors can purchase and use. Obviously, TMS is an extremely technical neurophysiological technique, so you need a fair bit of training, like with anything, to use it properly, so that you don't have false positive results or false negative results. Correct.
That's right. But it has huge potential, so that's actually very good and interesting.
Look, it has huge potential, and one of the things that we've applied this technique to a number of different questions in ALS. One of these is this phenomena of the split hand sign. There's an observation, an ever-burgeoning observation of dissociated muscle atrophy. The best recognizes this split hand, which was actually first recognized by Asa Wilborn from Cleveland, and then recognized by the Japanese and so on. And this refers to sort of wasting of the lateral group of hand muscles, the first dorsal interosseous. You can see here this hollowing of the hand. In actual fact, when Matthew Keenan was at the New York Yankees game, so Lou Gehrig was a New York Yankees player, he actually looked at his picture and he noticed that he had the split hand phenomena, which I thought was remarkably observant of him. And in conjunction, you have wasting of this thenar eminence of muscles. And there's relative preservation of the abductor digit minimi, the sort of the lateral hand group of muscles. And there's been argument as to what this causes. Some people think, well, it's specific metabolic demands of the anterior horn cell that supply this specific group. Others have suggested, well, it's the axons. And, you know, one of the tacks that we've taken is to sort of look at this issue of greater cortical representation. We hypothesize that because the thumb and the FDI has a greater cortical representation and thereby greater cortical motor neuronal input, that hyperexcitability in some way would contribute to this. And in a paper by our group, led by Professor Menon, we found that hyperexcitability was more prominent when you record over these affected muscles when compared to the less affected muscle. And this sort of correlated with biomarkers of lower motor neuron degeneration. So, you know, it can be used as a link, or, you know, could explain some of the symptoms that patients with ALS develop. And as we discussed, it also seems to have, or seems to be linked with patterns of disease spread. But I know you had some comments as well, Hande, and you've done some interesting research on this as well.
Yes. Yes, we have. And I don't know if you like me to share some of my slides, but before I go on to that, I also want to ask you a question, because this cortical hyperexcitability is a phenomenon we observe in ALS, but the upper motor neurons degenerate also in HSP and PLS patients. So do you have data to suggest that this hyperexcitability occurs, you know, only in ALS patients, or do you observe, or have you ever worked with HSP or PLS patients as well, and do you see the same phenomena in them too, or is it restricted to ALS?
Look, that's a very good question. The first thing is that cortical hyperexcitability is a dynamic process because it reflects what's going on at a cellular ion channel network level within the brain. And we know from animal studies that there are dynamic changes. The way that patients start in terms of their anatomy is not the way they end. And we know that there is an evolution of changes. In sporadic ALS, the typical ALS, patients tend to get some patients tend to get more hyperexcitable as disease progresses. But the other thing that we observed is that in the atypical variants, in particular the upper motor neuron predominant variant, or the upper motor neuron only variant, primary lateral sclerosis, you seem to have the opposite picture where you have inexcitability. And that's probably reflecting the degeneration of the output cells, that the cortical output cells that are seen on pathological studies. And these patients tend to have a much slower disease course and a sort of different disease burden to the sporadic ALS patients. They tend to survive for longer. But there is, you know, it is a dynamic process, and this has to be borne in mind when designing studies to perhaps look at factors that target mechanisms that mediate hyperexcitability. Perhaps these can be used to stratify patients, or hyperexcitability can be used to stratify patients into clinical trials. And also as an outcome measure, as we've recently studied with Brian Wenger and his group from the USA.
Definitely. So for this hyperexcitability issue, again, I want to come back to what you said, that it is dynamic. And then, you know, there are studies which suggest that there's first excitation, right? Overexcitation maybe, followed by silencing, so that they cannot be excited anymore, or their excitation levels are very low. But then it doesn't go back and forth, so that they are hypo excited, and then they go hyper, and then they go hypo, and then that doesn't happen. It's hyper, and then it goes hypo, and it stays hypo. Is that correct?
Yeah, look, absolutely. I mean, there are some indications that may happen. That at a cellular level, that there are dynamic changes in the level of hyperexcitability. Certainly, in some papers, hyperexcitability was shown to be prominent in the early stages and linked with neurodegeneration. And when you block this neurodegeneration with, say, potassium channel opening drugs, or even sodium channel blockers, but mostly potassium channel opening drugs to reduce the level of hyperexcitability. This seems to retard or slow down nerve loss. Conversely, others have suggested that perhaps a degree of hyperexcitability may be neuroprotective, and that as disease evolves, hyperexcitability occurs, and they've linked that to degeneration. I mean, that's a opposing view, and that's a matter of discussion. Certainly, I don't think it is our view. At an animal model, studies done by Avindra Nath, when they expressed or overexpressed the TDP43 gene in the HERV-K mice, or rather expressed the HERV-K gene in the TDP43 mice. They found degeneration in an ALS phenotype, and interestingly, they found cortical hyperexcitability in the early stages. And I know there are some studies that are expressing specific glutamate receptors in the animal model, and they found that when they do that through specific chemical means, they are able to stimulate, or rather reproduce, the ALS phenotype. Absolutely. But this is an extremely complex process. You know, at the one end is a loss of inhibition, but at the other end, there seems to be an increase in excitation. And another measure that we've looked at is this measurement called short interval intracortical facilitation. So you actually measure this by varying the size of your stimuli, your test and conditioning stimuli at the same time interval. So when you measure SICI, your conditioning stimuli is much lower. So it primes the inhibitory neurons, but it doesn't really stimulate anything. Whereas in this technique, we increase the size of the conditioning stimuli so that we can produce a response, both by the conditioning and the test response. And that's called intracortical inhibition. This is just the current required to produce a target response. It is below zero. And when we look at ALS patients, we find that in addition to inhibition being reduced, excitation or facilitation is markedly and significantly increased. This is an early process, and there seems to be a shift or a balance towards excitation, okay? So that in ALS patients, the degree of excitation is much greater than in healthy controls. And we're now studying to see how this evolves over time. These are extremely difficult studies to do. But interestingly, we found that this correlates with some functional measures, again, suggesting an association with functional disability. And this was work that was done by Mehdi Vandenbos in our group, whom we managed to attract from University of College London, and this is ongoing work. So cortical hyperexcitability is an extremely complex process. It is dynamic, and it is mediated by an imbalance of inhibitory and excitatory inputs, both at a network level, that is how the nerves talk to one another and the cortical output cells, but also at an ion cellular level as well.
That's right. Yeah, that's very true. Yeah, so we have actually collaborated with some groups in Serbia and also our colleagues here at Northwestern, and we looked at the extrinsic factors as well as intrinsic factors, you know, what's happening inside the cell, changes in gene expression, changes in the ion channel subunits, as well as their connectivity in the excitatory and inhibitory maps. So I would like to share some of them with you as well.
I might stop sharing my screen to get you, Hande, to sort of share your screen.
If I can share some of mine. Let me actually be here. Yeah. So this is actually one of the papers that we published last year, and it was in collaboration with Gordon Shepherd, Marco Martina, and Pavle Andus. And we looked at the electrophysiological determinants of upper motor neurons and how that contributes to their vulnerability. First, we looked at the their recordings within a large framework. We listened from the upper motor neurons, but there was actually excitation, field excitation, so that we can actually see which neurons or which sites they communicate with. And then, for example, they communicate mostly cells in the layer 5A region, and we can create their interaction maps. And so we did this in the wild type SOD, and imagine this is like P30, P60, a very early time point before there is massive symptom, and actually there's asymptomatic or early symptomatic. So this is actually very interesting because we looked at callosal projection neurons, which are very similar to corticospinal, but they do not degenerate in ALS. But they are born from the same progenitors. They migrate together. Some reside in layer 5. Some of them even, so close to corticospinal motor neurons or upper motor neurons, but they do not show vulnerability or degeneration in ALS. And if this is the callosal projection neuron, and if you look at their excitatory or inhibitory maps, there are really no differences. They are perfectly overlapping. But if you look at the corticospinal motor neurons, the inhibitory map, especially in layer 2-3, there seems to be an imbalance. So there is changes in how they are modulated, especially at the site of apical dendrites. So there are changes in the motor cortex that occur before neurons degenerate, before there's any symptom onset and so forth. And the other thing I want to tell you is we start looking at genes that are upregulated and downregulated, even show splice variations. And some of them are voltage-gated ion channels in their subunits. And, after we did all this study, we realized that there's, as you said, this is very dynamic. And there are players, both intrinsic players, which means these neurons have a different expression profiles of key voltage-gated ion channels and their subunits. But there's also dynamic change externally, so that the inhibitory neurons and their map is changing. And as you know, there are inhibitory neurons in layer 2-3, which modulate the inhibitory neurons in layer 5. So if there are changes in layer 2-3, then layer 5 inhibitory neurons are changed. So if they cannot inhibit the inhibition, you would have excitation. And I think this is also part of the problem that we observe. And as you said, this is a very complex phenomena. And I think if we understand this phenomena better, we may even use some of the FDA approved drugs that are used, for example, for cortex hyperexcitation in many other diseases. If we know when to use them at what time point, there may be off-target use for these FDA approved drugs as well. And I think, as you showed here, these are very important findings, and I believe that by studying the cortical component, we may even improve the clinical trial outcomes. And I think this was one review that you also authored. I see your name here. I want to ask you that. So by putting the cortex into the map, how do you think we can improve the clinical trial outcomes in ALS?
Well, look, thank you for that. I mean, it's a wonderful study, and I agree with just going back to the layer 2 and 3 interneurons. Modeling studies have suggested that the things that we're measuring in part are mediated by those layer 2 and layer 3 interneurons that project onto the output cells. Now, in terms of the trials outcome, and I think we've been unfortunately very unsuccessful in this, and there's a whole range of reasons for that. But one of the ways that perhaps we can use the cortex, whether it be hyperexcitability and or other measures of brain function, is to stratify patients into treatment trials. Most patients with ALS have accumulation of a protein called TDP-43, and there are studies now that have shown that you can measure TDP-43 in some of your biofluids. And people that progress more rapidly have a higher level of these biofluids. So I think this could be one of the ways you could use it, is to help stratify patients into treatment trials and use that as an inclusion criteria potentially. Obviously, we would have to make sure in terms of the cortical hyperexcitability that this is widespread and that people and that the investigators are trained in these techniques. The other way that potentially can be used is as a biological outcome measure.
Yes.
Most trials now use clinical measures such as the ALS-FRS and so on. I know people are going towards neurofilament and other biofluid measures. So cortical excitability can be potentially used as an outcome measure, especially in the early development of a repurposed drug, such as, say, retigabine, that we recently published on with with Brian Wenger and the group, to see whether or not there are any biological indicators to suggest that this drug may be effective, and that we can then take it to a larger phase 3 component. I think the FDA mandates that you have a functional measure. So I don't envisage that this will be used as a standalone measure, but perhaps in early development.
That's very interesting. The other question I want to ask you is, I remember your group was actually presenting in Dr. Kiernan, especially at ANA meeting, for potentially new diagnostic criteria for ALS. And I think in that study, there were suggestions made for new diagnosis criteria and so forth. So would you like to comment a little bit on that?
Yes, no, thank you. So the currently used criteria, the Ellis-Coriel and the Awaji criteria, are in themselves composed of various diagnostic levels of certainty, encompassing upper and lower presence of upper and lower motor neuron signs. And there are some issues with complexity, issues of patients, say, non-progressing from, say, what we call a possible category, which one would say, well, that's maybe ALS, but in actual fact, most people that have possible ALS do have ALS. And then some of the previous criteria perhaps would have included the atypical phenotypes, such as the primary lateral sclerosis. So in 2019, under the auspices of a number of organizations, including the International Federation of Clinical Neurophysiology and the World Health Federation of Neurology and so on, Matthew Kiernan and colleagues convened a meeting in Queensland, Gold Coast, for our annual or biannual neurophysiology meeting. And all the great minds, except mine, me excluded, were there. And we pondered on, well, what is ALS and how can we diagnose it in a simpler and easier way? And in essence, we came up with what is a novel diagnostic criteria, which I don't know if you've got another slide post.
Yes, I do. Yes, let me show you.
It reflects the clinical practice, and the importance of this is to identify upper and lower motor neuron signs in one body region, like an arm or a leg, in a setting of a progressive motor syndrome, and exclusion of all other things that could possibly cause this, which is how we practice. And that's enough to diagnose one with ALS or Lou Gehrig's disease or motor neuron disease. We also included a caveat of having low motor neuron signs in two body regions to encompass what was referred to as the progressive muscular atrophy form, which can, in a majority of patients or in a certain percentage of patients, progress to ALS. And important, we then discussed about, well, are there any objective biomarkers? And certainly for lower motor neuron dysfunction, we allowed the inclusion of neurophysiological criteria, EMG criteria, So, for example, when we put a needle into a muscle, we find specific features of motor neuron loss, and that was allowed as a surrogate or separate to just observation of muscle wasting and weakness. For upper motor neuron findings, we've left that flexible. At this point in time, it was agreed upon to predominantly rely on clinical findings, but should physiological measures like TMS and MRI and other fluid measures in the CSF become more widespread and more validated, then we've left the flexibility of including that in as a measure of upper motor neuron dysfunction. So in essence, to summarize, it's identifying upper and lower motor neuron signs in one region. If you've got that, you've got motor neuron disease. So this criteria says you've got it or you don't. And recently we validated that we studied this in a large cross-sectional cohort in Australia. It was a retrospective application of the criteria, and it showed to perform remarkably well. Obviously, we need further validation in multi-center cohorts, but we think that this will be robust and easy to implement on a wider setting.
Thank you so much. So I don't know if there are any other questions, and I thank you for your time for explaining us the importance of the cortical activity, excitation, and also the the silence of the cortex, and how this happens very early in the disease, and that we have to include the brain into the picture of ALS because it's a motor neuron circuitry that degenerates, and cortex plays a very important role because the movement starts in the brain, and this is a motor neuron disease. And the techniques, that the approaches that you developed actually helps us reveal how the cortex raises the red flag even before some of the symptom onset.
Look, thank you for that summary. Yeah, thank you for that summary. And just to end, I mean, really, motor neuron disease has been reclassified into as a dementia. And it's a great testament to Andrew Eisen, who initially thought of this way back in the 1990s.
As we had a webinar just like this one with Dr. Eisen, and my characterization was a little glib, I'm afraid, but I will use it again, and that all three of you were sort of into upper motor neurons before being into upper motor neurons was cool. And I had a question. I'm going to paraphrase a couple of the questions that came through the Q&A box, and that is that for ALS concerned patients or at-risk patients here in the U.S. is the cortical hyperexcitation tests that you did. And finding these inhibition levels and so forth, is that a test that patients should really demand from their physicians?
Well, I think they should discuss it with their physicians. I don't think demand is a good word, but
perhaps not. Maybe
I think they should discuss it. The threshold tracking technique itself is not yet not yet available in the USA. We are hoping to commercialize this, to bring it to clinical practice. We've already commercialized it, but to bring it to clinical practice so that physicians can use it. TMS itself has been used in a number of centers in the USA, and you've got some wonderful researchers there. And there is a form of cortical excitability that you can measure using the other technique. At this stage, that's, I think, on a research basis and not a routine diagnostic test, but we're hoping this will become a routine diagnostic test over the next 3 to 5 years.
And I think it's usually used within the context of spinal cord injury, and I see some spinal cord injury or rehabilitation centers who have TMS, but it's not been incorporated into the ALS fields because, again, as Doug said, the cortex was not cool, and then we didn't want to look at how cortex does or behave. But I think more research now, pushes us to that direction, that we really would like to listen to cortex, and if it is screaming or it is silent, and that actually tells us something about disease pathology.
Now, well put. Well put.
And I take that a step further, Hande. If patients are going through this process and haven't had the sophisticated tests like Steve is talking about, you mentioned that there were some FDA approved compounds that that could address this inhibition versus hyperexcitability and so forth. What would be the downside of prescribing those essentially off-label to patients now?
I don't think anybody can prescribe right now. We need to do the experiments, and first experiments need to be done in disease models, animal models, and then maybe collaborating with Steve or his center, or maybe some other centers in the United States or Europe. And but I don't think that it can be prescribed to patients immediately. First, we need to find out if drugs that approved by FDA, let's say for seizures or for cortical hyperexcitation, again to control the cortical excitation levels, if they have any impact on reducing the burden on either very active or inactive cortical neurons, and make the motor neuron circuitry back in balance. Because keeping things in balance is the goal. When the balance turns into imbalance, then vulnerability occurs. If we can keep the homeostasis, that's actually the solution. So if we can maintain homeostasis with electrophysiological manipulations, either small compounds or other ways, I think that would have some clinical implications, but I don't think it's going to happen tomorrow. Even this idea is an important step forward, because once we have the idea and we have the models, and we can look at the excitation and inhibition models and see if we can modulate them in the right direction, then next steps need to be taken. But at least you don't have to go to phase 1, phase 2, phase 3, because those drugs have already been proven not to be toxic and proven to have efficacy. So then once you have biological data, it will be much easier to convince clinicians or FDA that this may have off-label use.
Right.
Yeah, look, well put, Hande. I completely agree with that.
And I mean, just to borrow a baseball analogy, having previously been reviewed and approved by FDA kind of puts us on third base right away.
Yeah.
Is that okay?
I agree.
Okay. One other question that came up, I know, had to do with the applicability to PLS. And Hande, in one of your slides, I know that there was a PLS component, and I'm wondering if either of you could just say a few words about the applicability of these same hyperexcitability tests to PLS patients.
Well, I might take that. Well, in PLS, what we've found is that they are invariably inexcitable, and that probably reflects the underlying loss of nerves. Why that is important is because there are conditions that can look like PLS, and we're often stumped. Is it PLS or is it a genetic form of that disorder called hereditary spastic paraplegia? What we find in HSP is that the level of excitability is normal, surprisingly, which contrasts remarkably with PLS. And when you look at typical ALS, the degree of inexcitability is much higher in PLS than ALS. So the cortical excitability TMS techniques can help the physician decide from a diagnostic perspective.
Very nice.
Good. Well, I think it's probably a good time to sort of wrap up. I thank you both again so much for participating and for sharing some of this with us. I have to admit, the science can get a little bit naughty at some points, but this is super valuable for all of us. We're so appreciative. Just as a note to everyone, Kate FAHMS will be able to post this on our YouTube channel very soon, and I know that I want to watch it again just to be able to pull apart some of the details. But Hande, as always, thanks so much for hosting. Dr. Vucic, we just cannot thank you enough, and really encourage you to keep up your good work in upper motor neurons, because we're convinced that that's the key to the cure.
Yes, he not only keeps up the great work, I think he paves the way so that others follow in his in his path. So thank you so much, Dr. Vucic. Thank you so much.
Thank you for the kind words, and thank you for inviting me. It was an honor.
Thanks so much. Thanks, everybody.